Somehow, when we talk about toxicity of GMOs, we tend to forget there is an existing law, almost 100 years old, which has evolved over the 100 years, and that is called the Food and Drugs Act. Under that act, everywhere in the world, the company has to prove that the product, any product that is ultimately ends up in the human body, directly or indirectly, does not produce any toxicity. If it's one product, then it has to be tested that it's not adulterated by anything else, because that's how the Food and Drugs Act began, Adulteration Act. Then we have toxicity in animals, toxicity in embryos, toxicity for cancer. This is the existing law. Somehow, when we talk about GMOs, we think we have to go and prove, the public has to prove that these products are toxic. The law says the company has to prove that it is not toxic. So we went through this. I am a Health Canada regulator, and I hope to get back there one of these days. Because Dr. Hayden is here, I'm here. We were fired because we blew the whistle on RBGH. That was a genetically modified. And it was precisely the same thing that we're talking about now 12, 13 years later. That was the product. We raised the question, you have to prove that the product does not cause any toxicity in people. And we were told it's the same as the natural cow's hormone, so therefore don't ask any questions. When we asked the questions and we found that it actually produced severe damage, we demonstrated that it could cause cancer, it could cause antimicrobial resistance, it could raise all kinds of antimicrobial resistant bacteria. What happened? In Canada, we did not approve the product. Europe actually banned it. Now, just because the politicians are sitting here, I'd like them to pay attention to what I'm talking about. The effect of this, economic and health effects of this kind of science that is going on. Smart stacks are like the smokestacks. From these smokestacks are going to occur the same problem that we've had with the General Motors and the banks and all that. Now, India is going to buy it, now Europe is going to buy it, and our farmers are going to starve. Thank you.
I was wondering if you would foresee future regulations being imposed regarding the copy number, insertion size, or plant genome features at the insertion site. And then along with that, like the accompanying gene promoting elements that would be on the cassette or the vector.
Well, actually, if you look at the the plant guideline from Codex, there was all sorts of fights over that. But yes, they say there should be complete molecular characterizations, not only of what you're putting in, but what actually gets inserted. So it's, yes, copy number. It's also the structure of the inserts. They might be rearranged. So there should be a complete molecular characterization of what's actually in the plant, where it is, and looking at flanking sequences. And a number of countries don't do that. All they'll roughly do is see if the trait's there, but they don't do the complete molecular characterization as they should.
It's also quite interesting that if you look at the development of regulation, actually it has over time increased what is required. Because in the beginning, sort of like there was quite a relaxed understanding of what a gene is, of how genetics work. It was sort of much more sort of a sense of, oh, we can just use genes from anyone, put them anywhere, and we know what they will do. But over time, sort of the understanding has deepened, and also about epigenetics and other interactions, or about the mutation level which occurs. So therefore, if you look at regulation now, it actually has become stronger and stronger over time. So I'm quite sure the more we do research and the more we actually learn, we realize that we actually should require much more information. There's a problem at some levels when it said like, okay, we could get all this information, we could demand it from companies, and we would have like a complete sort of set of the proteins, a complete set of the metabolome of the genome, whatever. But what would we do with that stack of data? We don't know how to interpret it, which is quite interesting in a way that it's almost an acknowledgement that we do not know how to interpret it, and yet we clearly sort of aim then to simplify rather than to acknowledge we have more to understand and to learn and maybe go more the precautionary approach.
Yes, and then finally, I should just point out, when some of these crops were approved in the mid-90s, they didn't have the technology to be able to do a proteomic analysis or the chip array so they could look at gene expression. That only became available later. And that's why in the Codex document they talk about how there are things that can look for unintended effects, but they weren't developed until later, until they were able to be used. And so that's why now that some researchers in Europe are starting to do these experiments, they're indeed finding things. There have been a number of studies showing that for a number of the traits, the genetic sequence isn't the same as what was inserted. There's all sorts of rearrangements that have been happening when they look at the crops that are actually in the ground compared to what was approved. So part of what's going on is the science is getting more sophisticated so they can start to find these things. And then, of course, that raises the question of, well, when you did your original assessment, you didn't have the tools to look for things, so you just assumed that there was nothing there.
Yeah, a specific example in Canada was in 1996, we approved Roundup-Ready soy. And then in 2001, Monsanto sent a letter saying, oh, by the way, there was this whole other gene, a whole bunch of genes that were inserted accidentally, and we just want to let you know about that. Well, and this was raised recently to me as because I'd raised this previously on another panel, and a CFIA regulator said, well, yeah, but that shows the system works because Monsanto figured out that they had, that there was more going on, and they told you. Or they told us, and we assessed whether there was a problem with any of that extra genetic information or genetic material that went in, and we decided there wasn't, and so the system works. But it does beg the question if that's the kind of system we really want, that where we're discovering things after the fact and relying on industry to tell us that they've found something new.
And I would just point out that after Monsanto did that, a couple years later, there was another molecular assessment done in the European Union, and they found yet another set of changes that were not there originally and were not in the second submission Monsanto did. The US FDA, they have admitted, but they haven't finalized this rule. They did admit in 2001 that genetic engineering was unique, and that the question it raises is because of this insertional mutagenesis, because you're not controlling where you're inserting things, that causes all sorts of problems because they now realize that genes are in specific locations for a reason. And they said that separate data should be required on separate transformation events, and that they were going to be requiring data, but they, of course, have never finalized that. So unlike in Canada, where there's actually a formal assessment, in the US in terms of a food safety assessment, except for the flavor saver tomato, the FDA has never made a conclusion about the safety of any of their crops. All they've done is they send the companies a letter that says, we understand that Monsanto or AgroEvo or Syngenta thinks these crops are safe and don't raise issues that would require pre-market assessment. So technically the FDA has never made, and except for the flavor saver, has never made a conclusion about the safety of any of the foods. They just say that they understand the companies think they're safe and that they don't require further assessment.